Part 2, Methods. The X-ray crystallography process begins with Step 1, obtaining crystals, then Step 2, collecting X-ray diffraction data, Step 3, solving the phase problem, and 4, calculating the electron density map, and finally, Step 5, obtaining the 3D molecular structure. Step 1, obtaining crystals. In order to produce crystals, we must have purified protein. Like laborers in a factory that produce clothing, we can hire bacteria, yeast, or insect cells to manufacture a protein based on a gene template we give them. Because the target protein we need for crystallization is not the only protein produced, we must purify the protein. There are many different ways to purify a protein. The three main types are size exclusion, ion exchange, or affinity chromatography. For example, we can separate proteins by size. With ion exchange chromatography, we separate by the charge on the protein. We can use a negative charge resin to grab positively charged proteins, and positively charged resins to attract and separate negatively charged proteins. We can also use affinity chromatography with a tag bound to the protein, like a his tag, which can then separate tagged proteins from non-tagged proteins. The his tag, or another tag that is used, can be removed in a later step in the purification process. After getting the pure protein sample, we can start doing protein crystallization. Because we don't know the buffer conditions in which a protein is able to crystallize, we need to try tens, hundreds, or even thousands of different crystallizing buffer conditions. These conditions vary by salt, precipitants, and metals used and at varying concentrations. Basically, we need a plate like this one, which can be 24 wells or 96 wells. Each well has a reservoir where you can add a certain amount of crystallization buffer and 3 other smaller reservoirs to put the sample mixture with crystallization buffer. Once adding all the components, it is essential to seal each well properly. In principle, precipitant and crystallizing buffer would bind to water molecules and reduce the solubility of protein, which would lead to protein precipitation, or, if the conditions are correct, to crystallization of the protein. Step 2. Collecting X-ray diffraction data. You may be thinking, well, what makes a good crystal a good crystal? For every protein, the type of crystal that is a good crystal varies. The conditions in which the protein needs to crystallize changes, as well as the size and shape of the crystal. Ultimately, you want the best crystals of your protein to be used in the data collection process. Once you think you have a good crystal, you must prepare the crystal for the X-ray source. In order for the crystal to withstand the powerful effects of the X-rays being shot at it, the crystal is frozen with liquid nitrogen. This process also removes water from the sample, which can disrupt the diffraction pattern of the crystal if the ice is in the lattice of the crystal. By allowing the sample to be frozen, the sample can withstand the X-rays over a longer period of time. The crystal must be frozen quickly after it is mounted in order to withstand the powerful effects of the X-ray. The freezing is completed by plunging the mounted crystal in liquid nitrogen. The crystal remains in liquid nitrogen during the rest of the data collection process. X-rays are at about the same wavelength as a bond that is between two atoms, or about 1 angstrom. This makes X-rays the perfect source in order to obtain the molecular structure. Imagine if we use something different, for example, instead of X-rays. If we wanted to image a house, we could start with basketballs, which would provide only the large details or outline of the house. If we use something smaller like golf balls, we could then start to get the smaller details like where the windows and doors are. This same concept applies to X-rays for protein structure. There are different X-ray sources that can be used during experimentation, including synchrotrons or X-ray generators. The pros and cons of each are listed below. Here is an X-ray generator and a zoomed-in version of the X-ray generator itself. Here is a picture of the inner workings of an X-ray generator. The filament produces a stream of electrons which are attracted to the tungsten target. As the electrons move to the target very quickly, their energy is released as electrons. Here is a picture of a synchrotron. The concept of the synchrotron applies to electrons in order to produce X-rays. When an electron is moving fast enough, energy emits its X-rays. The energy is emitted when the electron changes directions. The electrons first begin to be accelerated in the particle accelerator before exiting into the different beamlines. The crystal is moved in a 360-degree pattern in order for the whole crystal to be imaged using the X-ray beamline. Think of one diffraction pattern as one piece of the puzzle. Each time the crystal is rotated, a new diffraction pattern is gained, which adds another puzzle piece. In order to get all the pieces of the puzzle, the crystal needs to be under complete 360-degree rotation. Then, the whole puzzle can be solved from the different puzzle pieces. In other terms, the diffraction patterns are solved and used to determine the final structure. Step 3. Solving the phase problem. You may be thinking, what is the phase problem? A light wave has not only an amplitude, but also a phase, which is systematically lost in measurement during X-ray crystallography. In order to account for a phase problem, different techniques have been evolved. MIR requires data collection from a native crystal as well as from crystals soaked in a heavy metal solution. MIR utilizes the diffraction intensity differences between the heavy atom derivative and the native data sets to obtain the actual diffraction pattern. SAD and MAD requires only one crystal, unlike MIR, but they differ from the number of data sets needed to be collected at multiple or single wavelengths. To understand molecular replacement, let's use a more simpler object, let's say a car. Molecular replacement requires the knowledge of a homology model. Molecular replacement utilizes the phases calculated for the homology model positioned in the crystal unit cell of the unknown protein. Step 4. Calculating the electron density map. Well, where do we go from here after we have our diffraction pattern? Well, the objective is to figure out the crystal structure from the diffraction pattern. So, what allows us to go from the pattern to the structure? The electron density map. This is an interpretation of the diffraction pattern as a plot of electron density versus the position in space. You can think of it as a two-dimensional slice through the electron cloud. The main thing to note about the electron density map is that they depict the general structure of the crystal. Remember that diffraction is caused by electron clouds. That means that the higher the atomic number of an element, the larger its electron clouds are. This is really similar to the topographical maps that they use on weather channels to show the height/size of mountains. The measured intensities are proportional to the coefficients of the electron density equation. And we also get phase by the phase problem. Then we can get the electron density by simply implementing Fourier transform. You may be wondering, what's the influence of the diffraction data and the phase? You will find out next. If we only have the low resolution terms of the diffraction pattern, we only get a low resolution duck. In terms of crystallography, there is a considerable loss of detail. At low resolution, your atomic model may reflect more what you expect to see than what is actually there. If a segment of data is missing, features perpendicular to that segment will be blurred. In crystallography terms, helices parallel to the missing data axes will become cylinders. Beta sheets parallel may merge into a flat blob. Beta sheets perpendicular to the missing data axis may be very weak. You could get into a lot of trouble with anisotropic temperature factors in this case. Let us combine the magnitudes from the duck transform with the phases from the cat transform. You can see the brightness from the duck and the colors from the cat. Using the magnitudes from the cat transform and the phases from the duck transform, you can get a complete object. Step 5. Obtaining the 3D structure map. After a re-refined electron density map with a decent resolution is obtained, an atomic model could be built upon that. The principle is very straightforward. It is simply docking amino acids into the density. Some amino acids have similar side chain density, but predetermined sequence of the peptide chain and corresponding chemical environment could help in identifying which amino acid belongs there. Model building is a trial and error process, and an experienced researcher knows tricks to speed up this process. However, the most crucial part is still getting a density map with a good resolution in the first place. No one could build a reliable atomic model based upon a map with a 20 angstrom resolution. There are multiple packages available to the research community for model building. One of the most popular is COOT, developed by researchers in MRC.